Hi guys, I'm Annie.
Hi, I'm Stephanie, and today we will be talking about the Lassa virus.
Lassa virus is a group 5 negative sense single-stranded RNA virus. It is part of the multinegavirus order and is in the Arenaviridae family of two-segmented viruses.
The Lassa virus was discovered in 1969 in Lassa, Nigeria. As you can see here, the arenaviruses are broken into two groups, New World and Old World. It is part of the Old World Arenaviridae viruses, given the virus is associated with Old World rodents. The rodents act as a reservoir who then infect the humans. A cross-section of Arenaviridae's virion shows grainy particles. These particles represent the ribosomes taken from their host cells. It is this characteristic that gave them their name, Arenaviridae, given arena is Latin for sand.
Lassa virus is a virus endemic to West Africa, particularly in Nigeria, Sierra Leone, Liberia, and Guinea. It is a leading cause of mortality and morbidity in the region, with 100 to 300,000 infections and 5,000 deaths annually. The Lassa virus genome has two negative-sense single-stranded RNA segments named L for large and S for small. The L segment encodes the viral RNA-dependent RNA polymerase and the small ring finger binding protein. Meanwhile, the S segment encodes the glycoprotein precursor protein and the nucleoprotein. The genome is considered ambisense as its genes are encoded in opposite directions. The Lassa virus virion is approximately 120 nanometers in diameter and features a round capsid and an envelope. The structural features of Lassa virus are extremely similar to those of other arenaviruses that cause hemorrhagic fevers, which include Machupo, Junin, and lymphocytic choriomeningitis virus.
The 7 steps of the Lassa virus cell cycle. Let's begin by explaining how the virus finds and enters the host cell. First step is for the Lassa virus to bind to the high-affinity receptor on the host membrane called alpha-dystroglycan, aka alpha-DG. This receptor is expressed in most of the developing and adult tissues and cells adjoining basement membrane. Viral entry then occurs once the pH level has decreased, and under acidic conditions the virus dissociates from its high-affinity receptor alpha-DG, and there is a receptor switch to LAMP1. Late endosomal entry factor LAMP1 binding occurs and facilitates viral fusion.
Replication and transcription. After injection into the host cell, the genome is replicated. L and NP proteins allow for the synthesis of full-length genomic and antigenomic L and S mRNAs. Next, the genome is transcribed. Genomic and antigenomic L and S mRNAs serve as templates for the transcription of viral mRNA. Next comes translation.
NP, LP, and Z are translated in the cytoplasm. Translated NP helps facilitate further transcription, translation, and virion assembly. GPC translation and cleavage of SSP from GP1, GP2 occur in the ER.
Lastly, we have assembly and excretion.
Z protein, in color blue, is the key regulator of virus assembly. Interacts with GP in red, NP light and dark green, and L orange to create viral ribonucleoprotein complexes into GP spike containing particles. This leads to the release of infectious virions from the plasma membrane of infected cells. ECRT helps transport Z to the membrane to help with budding.
Lassa virus causes Lassa hemorrhagic fever.
Transmission of Lassa virus begins with the reservoir, the Mystomys rat. It is a type of rat that enters and lives in human homes and food storage areas. The infected rodents excrete the virus in urine and droppings for a long period of time, sometimes until end of life. Transmission from rat to humans occurs through ingestion of rat or inhalation of particles. Human-to-human transmission can occur from exposure to virus in the blood, tissue, secretions, or excretions of a Lassa virus-infected individual.
Individuals infected with Lassa virus typically begin experiencing symptoms 1 to 3 weeks after exposure to the virus. 80% of Lassa fever cases are mild and include symptoms such as headache, fever, general weakness, and malaise. 20% of cases progress to have more severe features such as hemorrhage, respiratory distress, repeated vomiting, facial swelling, and pain. Complications of Lassa fever range from hearing loss to tremors to encephalitis to spontaneous abortion. The overall case fatality rate for Lassa fever is 1%. The case fatality rate for pregnant patients ranges from 7 to 30% depending on the state of pregnancy in which the patient is infected. Lastly, the case fatality rate of patients hospitalized with severe cases is around 15%.
Early stages of Lassa fever can be tested with RT-PCR. The later stages are tested with ELISA, which is more commonly used given it can detect IgM and IgG antibodies, in addition to the Lassa antigen. Only in highly controlled lab can the virus be cultured 7 to 10 days.
Ribavirin, a broad-spectrum nucleoside analog, has been successfully used to treat Lassa fever patients. This antiviral drug is most effective if given early in the course of the disease. Also important for recovery is supportive care that ensures adequate fluid and electrolyte balance, oxygenation and blood pressure, and treatment of concurrent infections.
There are a few ways that we can prevent Lassa hemorrhagic fever. One would be proper food storage. This helps avoid ingesting rat droppings in the case a rat does come inside the home. Second would be avoiding eating the rats. Third would be wearing proper PPE when treating LHF patients in healthcare facilities. Fourth would be sharing strategies on how to reduce rodent population in homes with those living in high-risk areas. Bye! Hope you enjoyed your project on the Lassa virus.